Yes. I asked just to dim the light a tiny bit because most of my presentation is in black. Thank you. That's perfect. You just have an idea. So if you have seen also my poster in the poster presentation room, I'm interested in, again, understanding how the nervous system, and in particular the brain that we have, evolved. And today I'm going to talk to you about like a sort of more side project of my PhD that is about the evolution of cell division in the nervous system. And I'm going to start by reminding you how the nervous system, as I was saying, is an incredibly complex structure. And we can see it if we zoom in. And one of the main reasons for this complexity is the incredibly high number of cells that are making the nervous system and the way that they're all interconnected. And another quite obvious thing is that these neurons, all these cells, form during development progressively. And during development, the structure of the nervous system is, of course, much simpler. And if we look again at how the nervous system developed in humans and in all other vertebrates, so all the other animals with a backbone, from fish to, again, reptiles, mammals, we see that it forms through a process called neurulation. So we start with a neural plate, so a single layer of neurons, which then bends and closes to form the neural tube. And a key feature of vertebrates is that throughout the development of the neural plate, the neural tube, the cells are proliferating. So they keep multiplying and so increasing the number of cell types, of cells in the brain. If we look at a section through the neural tube, we can see that we have these proliferative progenitors located here that keep proliferating, and then they stop proliferating, they detach from this apical surface here, and they by this process called delamination, and they start maturing into neurons. And this sort of process of proliferation and maturation happens at different rates along the nervous system, and this is one of the main causes for the regionalization of the nervous system. So here we see an adult human brain, and we see the different areas. We recognize the cerebral cortex in blue, the hypothalamus in red, et cetera, et cetera. The key thing that I'm interested for my project is that if we look through vertebrates, we see that despite the difference in size and shape, these regions essentially are the same and develop in a similar way, meaning that, of course, that the ancestor of all vertebrates already possessed a brain that was regionalized. And so the question is, how did this organization evolve? And to answer this question, we use Amphioxus, which is a marine organism that is very useful for evolutionary biology, mostly because of its key position in the tree of life. So we can see here that together with vertebrates, it forms a group called chordates, which are characterized by the same body plan, so the same way of building the body. And by comparing Amphioxus and vertebrates, we can reconstruct the features of the ancestor of chordates, and so how basically our body plan evolved. And Amphioxus is particularly good at this because it has had a slow morphological and molecular evolution. Here we can see a model of Amphioxus at the top compared to a model of a fossil chordate that lived 500 million years ago. We can see that the morphologies are relatively similar compared to us, for example. And so what we do is to study development of the Amphioxus nervous system and compare it with the development of vertebrate nervous system. Here we see successive stages of development. The nervous system is highlighted by the expression of this gene called ELAV. And we can see, especially if we zoom in the larval stage here, that the nervous system is very simple. It's made of very few, the number of cells is very low. But we also see that it's morphologically similar to vertebrates. In particular, it's still dorsal, so it forms at the back of the animal, like in vertebrates, and it has a neural tube with an anterior, though very, very tiny brain that we can see here. And so what I wanted to know in this side project was how proliferation influenced the development of the nervous system in Amphioxus, given that, as we've seen, it contributes to the regionalization of the nervous system in vertebrates. So first, I wanted to know which cells are dividing and are proliferating in Amphioxus during development. And so we used this double labeling approach in which we treated embryos with this molecule called EDU, which intercalates with DNA when the DNA is replicating, so it marks cells in the S phase. And then we fixed the embryos and looked at the localization of this protein called PHH3, which is present where cells are actively dividing. So what we end up with is an embryo with these cells that are proliferating marked by these fluorescent markers. We can then look through the nervous system and plot the positivity of for these markers along the anterior-posterior axis and do this for different stages of development and create this proliferation landscape. We can see here, again, the landscape, and here some selected stages of the actual embryos with the positivity of the markers. What we can see is that during the formation of the nervous system, proliferation is restricted to the anterior and posterior sides of the embryo. So it's polarized, basically. And only at the onset of larval life, the proliferation resumes throughout the neural tube. And what we wanted to know then, since we saw this curious pattern, is what is the fate of these cells that are proliferating. And so we used the pulse chase approach, in which we again labeled with EDU embryos, so we know where the cells are proliferating. But then we removed this, so we stopped treating with EDU. The cells that were dividing have EDU intercalated in the DNA, and we can see where they go. So we continue development, we chase, we could say, until the larval stage, and see where those cells that were dividing previously are. So here we see an example of the EDU pulse. So we see the cells proliferating, again, anterior and posterior. And then so the nervous system is here, dorsally, sorry. And here we see the chase, so where those same cells are at the larval stage. And we can see that posteriorly they are spreading throughout the neural tube, whereas anteriorly they are remaining concentrated in the brain. So the next question was, what are they doing, especially in the brain, since this was mostly what I was interested in my PhD. And with this technique, we also managed basically to birth date neurons, so to know exactly when neurons are born and when they're going. So we have neurons born here and their position in the larval brain. So what this means is that we can test the function of these neurons, basically, by selectively inhibiting proliferation at specific stages using this drug called hydroxyurea. So we block proliferation at a specific stage, and then we see how the development of the brain changes by looking at the expression of these of genes that we know are expressed in the brain in normal embryos with this technique called in situ hybridization, in which we have a target mRNA, which we have talked about a lot today. We can make a probe specific to that mRNA, which is tagged with a fluorescent marker that then, when it's beamed with a laser, it emits light. So it's fluorescent, basically. And so we have the fluorescence where that gene is expressed. And so if we compare normal embryos with embryos in which proliferation has been inhibited, here we have the brain in blue, and it's a bit tricky to see here, but we can see that the expression of selected markers, in particular this marker of neurons that uses the serotonin neurotransmitter and neurons that express this marker called OTP, are gone from the brain of embryos when proliferation is gone, whereas inhibited. Whereas, for example, neurons that uses glutamate as a neurotransmitter are not affected. So this means, again, that proliferation, of course, is required for the development of these particular neurons. So in conclusion, we have constructed this spatiotemporal map of Amphioxus proliferation. It allows us to birth date neurons, so to precisely see when neurons are born during development. This means that we can compare them better with vertebrates because we know when they are born. And this comparison shows similar processes. So as in vertebrates, proliferation is required for increasing the cell type diversity in the brain. But at the same time, we see also unique proliferation dynamics, in particular how this polarized proliferation during the formation of the nervous system. And this shows us how the tweaking, basically, during evolutionary time, the magnitude and also the extent, basically, of cell proliferation has been a key factor in the evolution of our brain, and of course, then in the huge increase of complexity that we see in the vertebrate brain and in our brain today. I'd like to thank everyone from my lab, in particular Toby, that has collaborated with me in this project. And of course, thank you all for your attention.
Thank you very much for that. I'm looking for some questions. By the way, I must say this slide was very nice. A party kind of feeling at the end. Any question from the audience or from the jury? We have a question here.
I'll have a go out of profound ignorance. I'm intrigued by the, as it were, just the, I don't know, the geometric similarity between the chordate and the vertebrate, and whether, as it were, simple linearity in a mechanical way induces these processes.
So there is, yeah, so the process of how the nervous system forms in, again, in Amphioxus and vertebrates is relatively similar. The mechanics is important in the sense that, but again, it's very much unknown in Amphioxus, of course, because it's much less studied than vertebrates. In vertebrates, we know that the geometry of the tissue is important, and also subcellular mechanisms, for example, movement of the nuclei, of the nucleus inside each cell during the cell cycle, so it moves sort of up and down, and this is very important for the cell to continue to proliferate and like overall then shape the nervous system. In Amphioxus, we don't know that much about the mechanics in the brain, mostly because it's hard to measure and study. These animals are quite tricky. For example, the larvae, they have cilia around their body that makes, and they start spinning during development. So it's very hard to like pin them down and do live studies, because if you stop them from spinning, like you block them with like agarose gel, for example, they basically destroy themselves. The cells, the cilia keep trying to beat, and so the embryo basically stops developing because the forces are tearing the embryo apart. So it's very hard to see mechanics. This is why most of the studies that have been done in this organism are on gene expression. So in which we can again see and compare the expression of particular genes. It's not going back anymore. There we go, went on in one. But we are trying in our lab to expand on this a bit and try to understand really this was my colleague Toby was doing, the physical properties of the embryo, basically. Well, yes.
Thank you very much. I'm looking for probably the last question of the conference. So if anyone has something to ask. Yes, please.
Are there different types of neurons that are developing, or do they split lineages later on? And do they all kind of work the same way? Do you know?
Yeah, I see. So let's say, especially again in vertebrates where we know this much more, when you look morphologically at the beginning, so the shapes of the cells, it's very homogeneous. So the neurons, the progenitors look exactly alike. Of course, there are already some, let's say, automated methods. So inside each neuron, molecularly, they are already different. So there is a sort of, like a innate, let's say, molecular difference that is guiding then the formation of the different neurons later in development, where we then can see morphologically that the neurons are different. There is also some external influences. So there are other tissues around that are secreting particular signals that are telling neurons differentiate into this type or this other type of neuron, or differentiate into a glial cell, which is a different type of nervous system, cells in the nervous system. But just by looking at the morphology, though, at early stages is very hard. In Amphioxus as well. If we look at the genes expressed, they are already very different, even when the neurons are not formed. So when the cells are spheres and not the classic like neuron with a nice axon that you can see. But if you look at the morphology, no, they're very similar.
Thank you so much, and the time is up.